Now, let us examine the case of lethal genes, which can be very well represented with the example of albinism and coat color in mice. Genes may affect viability as well as the visible traits of an organism. Sometimes organisms die when these alleles or genes are present in homozygous condition. These are called lethal alleles. The phenomenon of activity of lethal genes is called lethality, that is death. Lethal alleles can be either dominant or recessive and autosomal or sex linked. The gene involved is considered as an essential gene. Individuals carrying the dominant lethal allele will die for sure. Lethal effect of an allele may be dominant or recessive. If lethality due to homozygosity for a recessive allele, it is called recessive lethality. In this case, the phenotypic ratio is converted from 3:1 to 2:1. The lethal effect occurring in heterozygous individuals is called dominant lethality. Geneticists believed that mutation can alter only the appearance of an organism after Mendel's law were rediscovered. But it was discovered that a mutant allele can cause the death of an organism. When a specific allele or gene is mutated, it results a lethal phenotype. Lucien Cugnot is well known for multiple allelism at a genetic locus, and he is a French biologist who discovered lethal genes while studying the inheritance of coat color gene in mice. Lucien Cugnot discovered an incompletely dominant allele capital Y for yellow coat color in mice, which is fatal in homozygous dominant condition. When he crossed a yellow mice represented with capital Y with a yellow mice capital Y and expected the progenies in 3 yellow which is represented by capital Y, 1 white small y, but the observed ratio was 2 yellow and 1 white. Then he determined by this experiment that all the yellow mice were heterozygotes, that is capital Y, small y, and it was dominant over the white coat color phenotypic trait. However, no homozygous yellow mice were obtained. These premature dead forms are dominant homozygous. From a genetic perspective, this means that the yellow coat colors are heterozygous and the capital Y genotype is lethal in embryonic development. In 1910, William Ernst Cassel, who was an early American genetist, and Clarence Cook Little, an American genetics, cancer and tobacco researcher, as well as an academic administrator, established Cugnot's discovery of lethal genes by proving that a quarter of the offsprings from the crosses between the heterozygotes died due to the failure of implantation in the uterine lining during embryonic development. 1/4 of the offsprings, that is homozygous yellow mice, died, but Cugnot did not see in his tests. Studies of coat color in mice reveals how genes incorporate to determine the genotypic and phenotypic characters. According to Cugnot's findings, the gene capital Y has multiple effects. The gene capital Y controls the coat color of mice and affects individual viability. That means, the gene capital Y is dominant over coat color and recessive with respect to lethality, but the heterozygous yellow has genotype capital Y, small y, which somehow managed to survive. Albinism is an example for lethal factor in mice. 5 different types of genes interact to determine the coat color in mice, that is A, B, C, D and S. The gene A determines the distribution of pigment in hairs, The wild type allele capital A produces a phenotype called agouti, an overall greyish color or salt and pepper appearance. This is due to the band of yellow color on the otherwise dark hair shaft. The allele small a determines the non-agouti phenotype. The yellow band is absent, there is solid dark pigment throughout. The capital B gene determines the color of the pigment. The two major alleles codes for the color of the pigment. Capital B codes for the black pigment and small b for brown. The allele capital B gives the normal agouti color in combination with A allele, but gives solid black with small a. The capital A and small b genotype gives a striped brown color called cinnamon, and the genotype small a, small b gives solid brown. Now, the inheritance pattern of the A and B genes are as follows. When cinnamon is crossed with the black, or if agouti is crossed with brown, in the F1 generation you get agouti, and if agouti and agouti is crossed together, in the F2 generation you get the progenies in the ratio 9:3:3:1. 9 is agouti, 3 is cinnamon, 3 is for black and 1 is for brown. The capital C gene wild type is responsible for the color expression, and the small c gene prevents color expression. The small c genotype is epistatic to the other color genes lacking coat pigment are called albinos. Another allele of the C gene is the C superscript H, which is the Himalayan type. The C superscript H allele is the heat sensitive version of the small c allele. The CH is functional only at the colder body extremities and makes color pigment. It behaves just like the albino allele small c in warm parts of the body. The small c constitution produces a standard recessive epistasis modified ratio in the following cross. Now, we look into a cross between albino and brown or black with albino. In the F1 generation, the progenies are black. Now, when the black and black are crossed together, in the F2 generation, we get a progeny ratio of 9:3:3:1, wherein 9 is for black, 3 is for brown, and 3 again for albino and 1 for albino again. Genetic analysis of coat color in mice reveals that the distribution of pigment to the hair is due to the activity or interaction of many genes and where they are displayed as coat color. Different interactions between these genes can modify the color expression. Here we focus just one gene, the C gene, capital C gene, responsible for coat color in mice. The capital C gene produces the enzyme tyrosinase. This enzyme is active in the pigment synthesizing biochemical pathway which gives coat color in mice. Albino mice occur due to a recessive mutation of the capital C gene. The albino phenotype masks any phenotype that might be caused by the recessive homozygous small c. The capital D gene determines the intensity of the pigment of the hair. The genotypes capital D and capital D small d permit the complete expression of coat color in mice, but small d genotype dilutes the color and looks like milky. This is due to an uneven distribution of pigment in the hair. The possible coat colors are dilute agouti, dilute cinnamon, dilute brown and dilute black. This is another example of a modifier gene. Here the dilute black is crossed with brown or black is crossed with dilute brown, and the F1 progenies are black. When the black progenies are crossed between each other, we get F2 progenies in the ratio 9:3:3:1, the 9 representing the black, 3 representing dilute black, 3 representing brown and 1 representing for dilute brown. The capital S gene controls the distribution of pigmentation, that is, the presence or absence of spots throughout the body. The genotype capital S results in no spots, whereas small s is responsible for a spotting pattern called piebald in mice.